A very good day and a warm welcome to all of you good people, wherever you happen to be located in these COVID times. Welcome to this ASEAN Dengue Day conference, and in particular to the Symposium on dengue vector mosquitoes presented under the umbrella of the Resilience Against Future Threats, RAFT, a project headed by the London School of Hygiene and Tropical Medicine, of which Malaria Consortium is also a partner. So to all of you listening in, a sincere thanks for setting aside time to join in. So dengue and other Aedes-borne arboviruses are steadily increasing across our planet. And these arboviruses will for sure increase the impact as serious public health challenges to many millions of people. There is much we still do not know about Aedes vectors of some of these arboviral afflictions. In the three presentations this afternoon, we will have a peep through the window at some lesser known species that could play a role in transmission. We will look at the platforms for information sharing among global vector control practitioners. And we will also listen into the need to understand which age group of mosquitoes pose the greatest threat of transmission and therefore need a targeted attention. It's good to be aware of the differences in vectorial capacity, even within a particular individual. I would like to thank our three speakers for taking the time to share their insights and experience with us. But before I introduce you to our three speakers, I need to share some brief housekeeping notes to our audience. Please keep your eyes on the chat window to see important instructions and information which the conference organizers may post, or notes from other members of the audience. If you have a question, please place this inside the Q&A box. Not in the chat box. If you have a question, please put it in the Q&A box so it's all clumped together. It makes it easier just to find all the questions in one place. If you message someone privately or they message you, remember that unless you change the name at the top of the chat box, you'll be messaging them directly back again. So you need to change depending on who you're targeting your message to. Please also note that we are recording this meeting, which includes also all messages that are sent through the chat box, and this will be posted publicly later on. All right, enough chatter from me. Let's move on to our esteemed three speakers. These are going to be Professor Ronald Vargas from Mahidol University in Bangkok, followed by Dr. Frederik Seelig from the Global Vector Hub in London at the London School of Hygiene and Tropical Medicine. And finally, Professor Joe Lyons, also from the London School of Hygiene and Tropical Medicine. The three presentations will run back to back, one after the other, excuse me, and then we will have a joint question and answer session after that. So first up is going to be Professor Ronald Morales Vargas, and he's going to be talking to us about Aedes dengue vector species in Southeast Asia. Are there others than Aedes aegypti and Aedes albopictus? Let me just give you a quick rundown of Professor Vargas, who is an assistant professor in the Department of Medical Entomology, Faculty of Tropical Medicine, Mahidol University here in Bangkok, Thailand. He received his PhD in medical entomology from the Institute for Tropical Medicine at Nagasaki University in Japan. Dr. Morales specializes in biological systematics, taxonomy, ecology, vector-borne diseases, multiple disciplines. But his current research aims to ascertain mosquito virus ecology, identification, and characterization. He's also studying the vector competence of mosquitoes in domestic and peri-domestic habitats of human and animal dwellings. And his research targets viruses such as Zika, chikungunya, Mayaru, and dengue. So Ronald, I'm going to hand over to you, and we look forward to listening to your insights and research. Thank you.
Thank you, Leo, for inviting me to this very interesting meeting. So, as the title say, I will talk to you about some Aedes dengue vector species in Southeast Asia, but I will concentrate mainly in Thailand, where is the place where I am working, and where we have some other species that had not been involved or reported being infected with dengue or any other virus. So just as a part of introduction, I would like to show you this information. This is come from the Zootaxa journal, where they show several mosquito species around the world, let's say, accordingly the regions. For instance, in the Afrotropical region, Sub-Sahara Africa, South Pacific Island, and Australian regions. Also included the Oriental region that includes Southeast Asia and the America. That include the Caribbean island. As you can see, or maybe some of you are already familiar with all these species that appear in the screen. However, as you also may realize that we just know or concentrate our knowledge in Aedes aegypti and Aedes albopictus that are normally considered the most important vectors for dengue, and maybe some other viruses, arboviruses. So that is when we start to work with the mosquitoes that we collect in the field and that we are interested in Aedes species associated to dengue, or to maybe now Zika or chikungunya, the first thing that we see, or most of people see, is these two characteristics on the sputum. So that is say on the left, we will have, if this one has these characteristics, everyone will say, okay, this is aegypti. Then we'll see the another characteristics on the right. They will say, okay, Aedes albopictus. So that is what's unfortunately most of, or many times, many of us, including myself sometimes, we concentrate. So then finish everything there, and we continue doing any other biological information, insecticide, tests, and many things. So vector competence, for example, as well. However, there are some other characteristics that, and some other species that may share these characteristics, especially in this characteristic that we are saying Aedes albopictus. So that's why sometimes, most of the time, in my case, my particular case, I say albopictus-like when I see this. Because this one, Aedes albopictus, is belong to another subgenus that is called Stegomyia, that contain many other species in many locations. For example, in Thailand, we had around 17 different species that belong to this subgroup. So anyway, there are another characteristics that we should start to see. For example, the clipeos, that is this area near the antenna or the antenna. So if the mosquito had these scales here, the white scales, plus these scales separated here, so we can say, okay, this is Aedes aegypti. But to be honest, how many of those one working or doing entomology go up to that point? Only maybe they will go to that point only when the specimen has loose or cannot see these characters. So then as I say, we have these two characteristics, we can say Aedes aegypti. So if we go to the another side, in the side of the albopictus or the Stegomyia group, as you can see, this group doesn't have the scales in the clipeo. And then we should take a look to the Mesipemeron that is in this area, that is pointed also here with the red arrow. So then we can go to another characteristic that is in these scales or these bands in the abdomen. So as you can see, if we concentrate in the Mesipemeron here, there are separation that also is sharing with the sometimes with Aedes aegypti is possible to appreciate. However, this scale in the clipeo will help us to separate from aegypti and albopictus like. So then we should go to see another characteristic, as I said before, these scales in the abdomen. If the scale, according to some morphological scheme, they mention this, the position of this scale is upward, like here. So in that case, it's albopictus. We can say albopictus. So in the another case, if it is downward like this one, we should see another character, we can conclude is Aedes scutellaris. Of course, there is another characteristic that we have to see, we have to take a look in the last tarsomere of the legs that I did not include here because, okay, this is not a taxonomy lecture, and some of the speakers mentioned before sometimes also. Anyway, I would like to talk to you about this Aedes scutellaris. That's Aedes Stegomyia scutellaris. So I will talk to you, show to you some information that has been published, and many of the information is not published yet. So, but about the information that already published, a colleague here in this department of medical entomology already did this kind of work where, based in the phylogenetic analysis of the CO1 gene, she were able to separate these two species, albopictus and scutellaris here, and also include the Aedes reversi and Aedes flavopictus, and so she were in that time already exists this sequence of only one specimen. So which also is another species that's already been reported in Singapore, being vector competent for dengue viruses and also yellow fever viruses, as well as they report the same in Laos. So, and also here come back to the phylogenetic analysis, as you can see here, Aedes aegypti is completely separate very well. So, but may this claim from Aedes aegypti separate this claim where include albopictus, flavopictus that is in Japan, as I say, and also reversi, and coming close, scutellaris is highly predominant in the South Pacific Islands, some of them. So how we are now claiming that there is a scutellaris here in Thailand, that while our neighbors are saying that is malayensis, that they have Aedes malayensis that is very close to Aedes scutellaris. And in fact, in Thailand, in some recent morphological scheme for identifications, Scutellaris is not existing there. However, going much more back in the 1970s, 71 and 72, Dr. Huan report the present Aedes scutellaris and describe some specimens that were gathered during the mosquito Asia or Bangkok mosquito project, a very old project that, so anyway, also this colleague were further to study the shape of the wing, what is calling morphometric. So, and also doing that based only in the shape of the wing, she were able to separate these three species, Aedes aegypti, this is in the case of the male, and also albopictus and scutellaris. And here also the female separate very well. This is based on the specific landmarks in the wing. So, and also using another characteristic of the wing, the geometric of the contour of the wing. So she got the same findings. So this is support us our point that we had Aedes scutellaris in Thailand. So with one student, master students, I went also to study what the biology or the ecology of these three species in Thailand. So using a very simple methodology to collect, to set up ovitraps like this that I'm showing here. We select two areas in Bangkok, in the Bangkok province of Bangkok, and that these two selected areas are separated for around 6 kilometers in a straight line. So I select these two kind of containers as trying to mimic what sometimes we call it the human-made containers. Of course, this one are both made by humans, but this was plastic and this is a natural source. What is the earthen jar? So we set these ovitraps in some sub-districts of this area here. So, and then we monitoring weekly and to collect larvae for stage 4 and the pupa for more than 1 year. So this is what we found. As you can see here, the Aedes aegypti in the two different kind of jars. So starting from September 17 in the 2015 and up to August 29 of the 2016. So as maybe in many of the Southeast Asian countries, maybe this is a similar picture with Aedes aegypti. So, interestingly, here there is on May, which is just maybe when the rainy season in Bangkok is starting. So we don't have any sample, any positive container that's with larva and pupa. So what we did here is that larva and pupa, we take it to the laboratory and rear to adults. Then as we identify it as adults. So again, Aedes albopictus is in these particular areas is preferring to breed in the plastic containers, which it might be, for some people, might not be big news. So, however, then we do with Aedes scutellaris also. The interesting point in this data is that Aedes scutellaris, Aedes albopictus, and Aedes aegypti are breeding the same time of container. So without much competition, because for example, if you see here, where in this region, in this area that is called Talinchan, we found in that area only Aedes aegypti and Aedes scutellaris. No Aedes albopictus at all during the study period. And as you can see here, especially in the dry season, no any of these species. And before somebody may wonder, every time that we find the ovi trap with a few amount of water, we refill it with water from the area. And so to keep it, the container always with enough water for the mosquito be able to oviponer there. So as you can see here, we have some peak here from September to November, where the Aedes aegypti is very high, but also the Aedes scutellaris. This is in Bangkok. By that time, the rainy season already finished, and also is finished the transmission. It's about to finish the dengue transmission. So, and this Aedes scutellaris here, but no Aedes albopictus. However, based in some experience with working in here, for instance, these Aedes scutellaris have been identified as Aedes albopictus. So I tried to make some conscientization to my colleagues in Thailand that to pay more attention in these aspects. So what happened in the another region of what is called Banque, what is 6 kilometers away. So here, of which the area is a little bit more urbanized. So we start to see the predominance again of Aedes aegypti during the whole study period. And we had only a very few scutellaris in some very few terms, which coincidentally, in that area, we had Aedes albopictus as well. However, as you can see in these two graphs, I hope you can see, apparently Aedes albopictus and Aedes scutellaris don't like to stay together. Because when we had Aedes scutellaris here, for instance, very few times we can find it together in this in the earthen jar container. So, and when Aedes albopictus take the position of the breeding places, Aedes scutellaris completely disappear. Of course, this is larva and pupa monitoring, right? So again, at the end of the year, Aedes scutellaris reappear and no Aedes albopictus at all. So somehow Aedes scutellaris can live very well, go very well with Aedes aegypti, but no get along with the closest partner, that is Aedes albopictus. Of course, according to some old literature, they divided this Stegomia, subgenus Stegomias, divided in two groups. In the group albopictus and scutellaris groups. So that is very old literature in taxonomy. So we also study, before the next point, I would like to show you that when we comparing, or we try to associate, because that was one of the objectives, to associate the predominance or the presence of these vectors with the dengue season, dengue incidence. Okay, so as you see, can you see here, there are the two species here during the high peak of dengue. So still define which species, any of these two species might be possible to transmit dengue. So that's why it's another part of the study that I have not been able to complete yet. So which is to determine the virus presence on those mosquitoes that emerge from those larvae and pupa. Of course, here it will be depend of the characteristics of the transovarial transmission of the dengue viruses in any of these species. So in this area, we also had albopictus. So, but as you can see here, even the dengue incidence is very low. Albopictus is still there. So in the another area, okay. So, one of my slides is lost. Okay, so it seems like this is my last slide. So the only thing that I would like to send to all the audience is that we should be more this data has demonstrated the value of careful taxonomic studies before undertaking any biological control or ecological, and when feasible, disease epidemiological studies. So this is my last slide. So I will very happy to entertain any questions when the session for questions come out.
So, Professor Vargas, thank you so much for that very enlightening presentation, where it's made a lot of us who work on dengue vector ecology and dengue epidemiology, made us sit up straight, realizing that what we often assume to be albopictus may not necessarily be albopictus, and we need to double check and make 100% sure of the identity of the species we work with.